So I'm recording now. Please keep your microphones off if you don't want to be recorded.
Hi, everybody. Welcome to our Ridge webinar series to this lovely summer day talk. It's with great pleasure that I'm introducing Rachel Boschen-Rose. I met her when she was a graduate student on a workshop in Galway, where we had a very nice chat. And then she moved to New Zealand, where she has done her PhD. She was also there a guest lecturer at, then she moved to Canada to do a postdoc at University of Victoria. After a postdoc, she was a senior project officer at the Seascape Consultant. So it's one of the first scientists that moved to a private company that I know. And now she's a visiting fellow at the University of Southampton at the Oceanography Science. And she also found her independent consulting trading as Boschen-Rose Ocean. So she also an entrepreneur. And she's going to present the work that she has done with her colleagues from New Zealand about the ridges and associated seamounts from New Zealand and the knowledge acquired towards conservation. And without further delay, I will give you the word. Thank you so much for being with us. Rachel, the floor is yours.
Thank you very much, Anna. And thank you everyone for joining this talk this afternoon. I guess good morning, good afternoon, and good evening, depending on where you are in the world today. So without further ado, the first thing I'm going to do is what people normally do at the end, which is to acknowledge some people who've been particularly important towards the research that I'll be talking about in this presentation. So I wanted to say an early thank you to Ashley Roden, Malcolm Clark, and Jonathan Gardner. As Anna mentioned, I started my deep sea New Zealand research in 2012 when I moved to New Zealand to do my PhD. And Ashley, Malcolm, and Jonathan were my PhD supervisors. So a lot of the deep sea mining related research that I'll cover in this talk was conducted as part of my PhD, and then later through continued collaboration with these three wonderful people. So I wanted to say an early thank you there. In terms of the structure of the talk for this afternoon, there are sort of 5 broad areas that we'll go through. The environmental setting of New Zealand, including the location, a little bit about the tectonics and the oceanography, some of the biological communities. I'm going to focus on seamounts and hydrothermal vents, the maritime activities that are going on within deep sea of New Zealand, and I will focus on fisheries and deep sea mining there, the environmental impacts of both of those activities. And then lastly, we'll delve into some environmental management, how these activities could be conducted and how conservation measures could be implemented in New Zealand. So firstly, the environmental setting in terms of location. I discovered when I was in New Zealand that there are many, many maps of the world, and a lot of them have New Zealand right off in the corner. Some of them somehow managed to cut New Zealand off completely, much to the chagrin of my colleagues in New Zealand. So I thought for once we'd have a map where New Zealand's in the middle, and that's what we have in this first image here. And you'll notice that New Zealand there's a lot of blue water around New Zealand, strangely enough, and the closest sort of large land masses are Australia, and Antarctica is actually not as far away as you might think. So we think of New Zealand as being isolated, and in a terrestrial sense, it is. The landmass is really quite isolated. But if you look at the image of the globe here, I presume you can see my mouse, you'll see that underwater New Zealand is connected. So we go up a structure here. This is the Tonga Arch and sort of arc, sorry, and sort of ridge system, which we'll talk about a bit later in the presentation. And then we also have connections south, and this influences the kind of biological communities that we get in New Zealand. Now, if you zoom in on New Zealand, then we can start to see some of the features that will come up again in this talk, and we'll come back to similar maps so you don't get hopefully too lost as we make our way around New Zealand. And we also have a little bit of reference to different biogeographic regions, sort of the north, the south, and the central area, and that will come in a bit more in the oceanography. So in terms of the environmental setting, the first thing we're going to tackle is tectonics. A lot of you may know New Zealand for being, particularly in this series, as having volcanoes and earthquakes and all sorts of exciting things. And that's because we have a couple of plates that are colliding. So we have a subduction system where we have the Pacific plate is slowly going underneath the Australian plate here. You can see that in the main diagram. And you can also see it in this sort of divided diagram where you can see a little bit below the surface. And this tectonic activity is part of the reason why we have some of the seafloor structures that we will discuss a little bit later in the presentation. So the seamounts, which we'll be talking about a lot today, are partial. Some of them are resulting from that tectonic activity, and some of them are not. So we have seamounts in different settings. This is a global map of seamounts, large ones, intermediate ones, and small ones according to the different colours. And if we zoom in, I'm sorry it's a bit fuzzy, but if we zoom in on New Zealand, again, you can see those kind of tectonic boundaries, and you can see several areas quite prominently with seamounts. There are lots of seamounts around New Zealand. And this particular in red, this area here is the Louisville Seamount chain. It's on the Pacific plate, is actually very old. So this is not forming as a result of tectonic activity. The Louisville Seamount chain is gradually coming this way and actually being subducted. And if you go along the seamount chain, some of these have cobalt-rich crusts as a mineral resource, but we won't be talking about them much today. If we look in a bit more detail about some of the seamounts within New Zealand and their distribution, there are lots of different depth ranges of these seamounts, and you can classify them accordingly. But within the exclusive economic zone of New Zealand, there are 718 seamounts. It's an inordinate number of seamounts, and many of these have been fished, which is something we'll come back to later. So 161 of those have been fished with at least 5 toes or more. Now, the tectonic environment definitely is what is responsible for our hydrothermal vent systems, and we're going to talk a lot more about them today. So again, you've got these features that form due to the tectonic activity. You've got Kermadec Ridge, and here you have a number of seamounts which were formed directly as a result of that tectonic activity. And some of them are hydrothermally active volcanoes, which are the red ones, and some of them are highly inactive volcanoes, which are the yellow ones here. And I'm just going to zoom in on one example volcano, which we've studied fairly well, Rumble 2 West. This is what some of them look like. Obviously, each seamount looks a little bit different. But when we say that it's hydrothermally active, we don't mean that the whole thing is giving off hot fluid everywhere. It's quite often only in patches. So in this case, it's a small area up here called Proteus 1, which again, we'll come back to a bit later. And then if you zoom in again on Proteus 1, you can see that there's distribution of different types of hydrothermal activity within that site. And you can see it's really not a very large area there. You have hydrothermally active chimneys, inactive chimneys, active mounds, and inactive mounds. And here's an image of what one of those active chimneys looks like. So continuing with the environmental setting, I also want to do a little bit about oceanography. I'm not an oceanographer, I'm a biologist, so I'll apologize in advance if I don't quite get this right. But there are a couple of different levels of circulation that I thought we should have a look at before we move on with the talk. And the main take homes, I guess, in terms of the surface circulation is you have some warm water that comes in from Australia. As I said, the proximity to Australia and Antarctica is important in terms of the New Zealand environmental setting. And we have cooler water that comes in as fronts from Antarctica. And you can see some of the patterns of circulation here, and there are some eddies up in this region. And this here, just for orientation, is the Kermadec Ridge, Colville Ridge, General Kermadec Arc structure. This here is somewhere called the Chatham Rise, which we will talk about later. If we go deeper, the circulation patterns change. So you don't see that warm water that comes across here, although some of the eddies that we see in the surface circulation can extend down to 1,000 meters and influence the sort of water structure down here, which is what you see, these little circles. And generally, the sort of deep sea environments that we'll talk about in this presentation are deeper than 1,000 meters or round about 1,000 meters. So it's this level of circulation that's potentially of more interest. So now we move into the biological communities. And I wanted to start with seamounts and so sort of some general seamount community information. So on the right, we've got sort of illustrations of the types of organisms that we often find on the hard substrate or slightly sedimented substrate of New Zealand seamounts. And the elevated hard substrates are typically dominated by coral and sponge assemblages, which is what we see elsewhere in the world when we look at seamounts. If we were to characterize all of the organisms that we see on different video transects and then call those transects a sample, we could then determine what communities or assemblages that we find within those samples. And we've done this in a couple of different studies. And what we can see is that different assemblages. Each of these little coloured dots is an assemblage in a sample on the seafloor, and so assemblages that are the same are all the same colour. And any assemblages that have a little black dot in the middle are only found on that seamount. Any assemblages that are just, you know, an unfilled circle are found at different seamounts. This is a 3-mount study. So what we can see is that some assemblages are shared amongst different seamounts, and some only appear to occur on one seamount. And when we looked at some of the environmental variables that might explain some of the distribution of these assemblages, hydrothermal activity was actually the best predictor of assemblage structure at the seamount scale. So just for context, Brothers is hydrothermally active, and at the time of survey, Rumble 2 West was believed to be hydrothermally inactive. So continuing with the biological communities at seamounts, I wanted to do a little bit about connectivity. Seamounts have often been called islands in sort of the deep sea mountains, underwater mountain ranges. And because of the differences in depth, seamounts are not directly connected. So if organisms are going to move between seamounts, then they often need to move via currents as larval stages. And I'm sure you'll hear more about that from your speaker in the next Interimage series. So, and different taxa we found when we've looked at this in New Zealand, they have different connectivity patterns. So from a management perspective, that can be challenging. So there's different genetic differentiation varies amongst taxa. So patterns of high levels of genetic diversity, for example, can be different for different areas, for different species. You don't always have one seamount that is really important for genetic connectivity. It could be different seamounts are important for different species, or even different parts of the same seamount are important for different species. We have differences in effective population size, which is the proportion of the population that contributes to reproduction. And where you have very low effective population sizes, it means that you have few individuals reproducing and are potentially more vulnerable to disturbance. And different seamounts could be important as source populations. So the populations that contribute most organisms to, most young, most larvae, most recruits to the general population. But for many of the taxa that we've looked at and that my colleagues have looked at, the Kermadec Ridge is an important feature when you put it in the context of other features in New Zealand and beyond. So those Louisville seamounts that I talked about, for example, this is the Kermadec area, the Kermadec Ridge here, the Chatham Rise, which I mentioned in the beginning, Macquarie Ridge, and also Campbell Plateau. So I've got one example species here, Desmophyllum dianthus, which is a solitary stony cup coral. And population connectivity work for this species, the Kermadec's population is distinct from Chatham Rise, and you can see that in this diagram because if there's a sort of a trail, I guess, that goes from one color to another, then that means that organisms are traveling in that direction, larvae are traveling in that direction. If there's no connection, then they're not traveling between them. So you can see that there's no link from the Kermadecs here to the Chatham Rise. And there's also high levels of self-recruitment. So that's shown by this hump. That hump means that there's self-recruitment. So organisms at that site are releasing juveniles that then recruit back at that site. They're not dependent wholly on getting young larvae recruits from elsewhere. And what this diagram also shows that the Kermadec region is a very important source population for Louisville seamounts, which again, we mentioned earlier on in the presentation. Additional work has demonstrated that those shallower populations are distinct from deep populations. This is something we see for other species as well, not just this particular coral. And it means that connectivity doesn't necessarily happen from shallow to deep. That's very unlikely. It's more likely to happen from one deep area to another area at a similar depth. And that is facilitated by the ocean currents that we looked at earlier in the presentation. So the next biological community that I'm going to introduce you to are sort of hydrothermal vents and hydrothermally active areas. So zooming out from New Zealand a little bit to look at the global scale, scientists have done biogeographic studies to try and see if there are regions in the world that have similar types of hydrothermal vent communities, and they can divide the world up into these biogeographic regions. If we zoom in on New Zealand, which is where this box is here, you'll see that they have a dark green color that we don't see anyone else. This is the Kermadec Arc region. So it's a separate biogeographic province. The hydrothermal vent communities that we get here, we don't see them quite like that anywhere else in the world. And I've got some pretty pictures here, because who doesn't like some pretty pictures of some example organisms that we find there. So hydrothermally active areas where we have hot water coming out of the seabed with its various chemical soup. We have very sort of hot hydrothermally active areas. This is on Brothers Seamount, and you can see it's colonized by some shrimp in there. We also have diffuse venting areas where its temperature's not as hot and there's not as much water coming out. So this is a little collection of Gigantidus gladius vent mussels, and you can see that they're being predated upon by the sea star, much like we see in the shallow rocky shore, where you get sea stars preying on shallow water mussels. We have another mussel species that we find in the Kermadec Arc called Bacchimodiolis monacensis, which is found elsewhere. So Gigantidus gladius only occurs on the New Zealand Kermadec volcanic arc, but Bacchimodiolis monacensis also occurs within the Manus Basin, strangely enough. That's where its name comes from. And we also have a very cool stalked bent barnacle. I don't know how well you can see it in this image, but the feathery bits here are what it uses to gather up its food. And you can see that the crabs here are also gathering up it for their food. But as well as having distinct species on hydrothermally active areas, we also have unique assemblages on hydrothermally inactive areas, according to some of the work that we've done. So once upon a time, this was an active hydrothermal vent chimney, which has stopped venting. And it stopped venting well over 100 years ago, which we can tell from the age of the corals here, the size of the corals that are growing. So this particular coral, which we think is Selinosmilia variabilis, which I've got that wrong there, sorry, it should have been Selinosmilia, and Dermachinus horridus urchins, we find these elsewhere within New Zealand and further afield. So we don't have unique species in terms of hydrothermally active habitats, but we have unique assemblages. So the way that components of that community come together and the abundances of different individuals. Again, sticking with hydrothermal vents, we also have a relationship between the biology that we see here and also the mineral deposits that are of interest to deep sea miners. So the seafloor massive sulfide deposits that they're interested in form through hydrothermal activity. Hence, where you have a sulfide deposit, you often have hydrothermally active communities. So those sort of endemic organisms we looked at in the slide before. And we can see that here we have different assemblages here, again, different colors of different assemblages. When we look in detail, we can see that the hydrothermally active endemic fauna occur in the same region as these hot colors, which is where those particular assemblage types are. And then we can map that against the known hydrothermally active chimneys and mounds and the inactive chimneys and mounds. And what we also see, so this particular outline, this is Proteus 1, which I mentioned in the beginning, and it's off the flank of Rumble 2 West here. This was a site that was provisionally licensed for prospecting of deep sea mining. So before they go in and actually dig up the seabed, they need to go in and see what sort of mineral resources are around and start to characterize the environmental baselines. So this was an area that a company called Neptune was looking to mine. So we were trying to see what lives there and whether the defined reference site was suitable or not. And you can see that hydrothermally active areas here occur in very close proximity to hydrothermally inactive areas. It's a real mishmash. The whole site isn't active in that sense. And both of these host unique assemblages. And again, when we looked at environmental variables, hydrothermal activity was the best predictor of assemblage structure. Now, you hear me talking about assemblage instead of community, and I should probably explain why, because it will come up a bit later in the talk. A biological community is everything that lives there, from microbes through benthic invertebrates or seafloor invertebrates, all the way up to fishes. Whereas it's very challenging to characterize an entire community. Quite often we only characterize part of that community. So for example, the large seafloor invertebrates we can see from imagery, as was the case for this study. And in that case, we call that subset an assemblage. So it sounds pedantic, but it's important, and we will come back to it a little bit later. So again, more biological communities and hydrothermal vents. I wanted to give you a little bit of connectivity pattern background as well. So we looked at this for seamounts, and our example there was the cup coral. Here, the example is Gigantidus gladius, my favorite vent mussel. And what we can see from this map, I should probably explain what this map actually shows. You've got New Zealand here, and this is all along sort of the seamounts of the Kermadec Arc, particularly the Kermadec Bridge. You have these circles, and the circles are populations. So within these populations, different colors are different haplotypes, so different versions of the gene. And you can see that some populations, like this one in the middle, have lots of different colors. So they're very genetically diverse. They have lots of different versions of the same gene. But if we look up here, we can see that there are only 2 versions of that gene. And in fact, if we look here, there's only 1. So that's broadly what that map shows. And here we've got some images of Gigantidus at the seafloor. And this is just to illustrate that this particular mussel lives in a variety of habitats. So here it's on the edge of a fairly hot venting area. And you can see some of the different minerals here. And then we have Gigantidus in a more diffuse area. And then we have it here in a mildly sedimented area. So it's a very adaptable mussel. It can live in a lot of different hydrothermal habitats. And it also colonizes a rather extensive depth range. Having said previously that for that coral, populations at different depth ranges aren't connected. Somehow populations of this particular vent mussel are connected, and say there's something like hundreds of meters of range there, which is unusual. And what we see in general from the tests that are behind this particular plot is that there's very limited spatial genetic differentiation. So populations are all quite similar and they're all quite well connected, what the tests say. However, if we look at this one here, population 3, there are lots and lots of colors in that particular circle. In fact, that's the most number of colors in any circle on this particular diagram. And that's at Rumble 25 Seamount, which is the most diverse population. And there is also very high migrant exchange. So recruits or larvae, sort of young organisms coming into the population and also coming out of that population to other populations. So we thought at the time this may be a source population, which helps maintain the population connectivity for other populations on the ridge. So it's potentially an important place. Coming back to our oceanography again, you remember I mentioned the eddies on the arc, and I think they're somewhere around here, those eddies that I was talking about before. And when we wrote this paper, we hypothesized that the East Cape eddy was actually constraining larvae within this central part of the arc here, where you have these very diverse populations. And that's why, part of the reason why this population looks so different and has such low genetic diversity. And the mean current flow is also southwards at 1,000 meters, which is, as you'll see, roughly the kind of depth we're talking about for Gigantidus gladius. So most of the larvae transported in the water column are probably headed this way, not that way, to sort of replenish this population. So now we're going to take a step back from the biology, and we're going to look at some of the activities that go on within the New Zealand EEZ, and then we'll move from there into environmental management. So we're going to start with deep sea fisheries. So the map on the left shows all of the existing protected areas or sort of closures and measures. And you can see we have the red boxes are benthic protection areas where all bottom trawling and dredging is prohibited. So from a fisheries perspective, we also have seamount closures, these green areas here, where there's a prohibition just on trawling. And this area over here, the sort of the AFC and AFC up there, these are Australian fishing zones, and they're managed by Australia, not New Zealand, as far as I understand. So where we were talking about Gigantidus gladius, that mussel was sort of up in this region here. And in terms of fisheries, we'll come back to this Chatham Rise area a little bit later in the talk. I also wanted to point out this bit here, which is the Kermadec, and this was actually a benthic protection area in this map, but it also has some revised status that we'll come to a little bit later in the talk. And I just wanted to point that out here. You can also see the concentration of fishing activity within New Zealand territory. So that's what the second map here shows. And in this case, hot colors, so sort of red, yellow, and light greens show higher intensity of fishing, and the very sort of gray, blue type colors here show very low intensity of fishing. And you can see here that the Chatham Rise feature that I've mentioned a couple of times has a much higher concentration of fishing than the Kermadec volcanic arc area up here. So moving back to the Kermadec volcanic arc, we've just seen there's not a whole bunch of fishing up there, partially because there's this very large benthic protection area up here and also tectonic reach benthic protection area down here. Although they don't have fishing activity as much in this region, we do have, or we did have, potential deep sea mining. So this would be for seafloor massive sulfides or polymetallic sulfides, depending on which name you prefer. And those are the mineral resources that form through hydrothermal activity. They're typically rich in this region, they're rich in gold and silver and other metals. So you can see in this map, you've got the dark blue here. These were prospecting licenses issued to Neptune in 2002. So that very early map I showed you with that sort of squiggly looking site, and I said that there was hydrothermal activity distributed in different parts of that site, Proteus 1. That was an area licensed to Neptune Minerals. So that's actually roughly here on that map. You can also see a lot of the seamounts, the known seamounts, red, hydrothermally active seamounts, and yellow, hydrothermally inactive seamounts. And then and you can see here as well, this hashed area, which is the same as this orange area in this map, is the proposed Kermadec Rangitohua Ocean Sanctuary. This is something that was proposed in 2015 by the New Zealand government. It's not yet enacted, and we'll talk a little bit more about that later. But it basically sits over the Kermadec benthic protection area. One of the newer developments is this offshore reserved area in pink, which wasn't available in this map at the time, which is why I've got a duplicate map here. And in this particular location, no new mineral license applications are considered. So the Neptune permits have long since expired. They're technically on hold, so they're still available if Neptune wanted to come in and continue their work. But no new or additional permits can be granted in this entire region until the New Zealand government decides what it wants to do about deep sea mining. So now we're moving into environmental impacts. Having talked a little about the environment and some of the activities that are going on, we're going to start with deep sea fisheries. And we're going to hone in on, again, the Chatham Rise, and a group of seamounts with very amusing names, Morgue, Gothic, Zombie, Diabolical, Ghoul, and Graveyard, and we will focus on Morgue. So there've been a number of studies on looking at seamounts and fisheries activities in New Zealand and what the impacts are. I'm just going to focus on a couple of those, and very recent studies, in fact. So Morgue, this seamount here, it was closed to trawling in 2001, and there was a time series study that was done, and there were very little signs of recovery by 2015. And when they looked at, they put out some recruitment settlement type plates, and there were no signs of recruitment after 5 years. So if you look at this metal disc here, you'll see the little yellow boxes. Those are recruitment plates that were put out, and there was no sign of recruitment after 5 years. This is Morgue Seamount here, and this is what they were fishing for at Morgue. So this is an echogram of the seamount itself, and then these areas here at the top, and then just off to the side of the summit are Oceanography aggregations, which you can see in panel B, which were a target species. And if you look underneath them, you can see there's all this coral rubble from where people have been trawling and damaging the coral. And if we look at the bottom two panels, you can see on the left in C what the seafloor should look like if it hadn't been trawled with your coral. So then the Smiler variabilis and some really nice really nice stars, other sea stars, different corals. And this is what it looks like when it's been trawled. So all of that is gone, although some orange roughy are still hanging around. Continuing with deep sea fisheries and environmental impacts, as a recent paper that sort of tried to dissect this a little bit and using data from New Zealand seamounts and some other seamounts. So you start off with your undisturbed benthic communities. That was the image that I showed you of the nice corals. And then you have a trawl that comes through the middle and effectively damages or removes whatever was in the path of the trawl, although the level of impact depends on what community was there in the first place and all these various caveats. As time goes on, large organisms, adult organisms or larvae may come in and colonize, and this is where your population connectivity is important. And if you have successful settlement, then you may begin to see signs of recovery and then into growth. But as you can see in this image, the pre-trawling community here and here is still very different from the post-trawling community in the middle. So this particular paper, their main conclusions were that bottom trawling impacts can last for decades. Seamount communities have very limited resilience to trawling. That's partly because they're very slow growing, or some of the organisms are slow growing, long lived, such as corals, and also quite fragile. And that the potential for recovery from trawling varies amongst the different taxa you see at seamounts. If we look then at deep sea mining, the other activity we're focusing on for this talk, it's important to say that seafloor massive sulfide mining hasn't happened anywhere in the ocean, in the global ocean, in fact, at least at a commercial stage. But from what we understand about the technologies that are being developed, it's expected to have impacts from surface to the seafloor. And that's what this diagram shows. You've got your mining vessel up here. You've got a crawler on the seafloor, chipping away at the seafloor and then pumping all the mineral up to the surface. And then the wastewater is returned back to the seafloor. And this here is the seafloor massive sulfide deposit that they're actually mining. And this tiny seafloor production tool here, this is what they actually look like. They're not tiny at all. So these are the full-scale versions that were developed by SMD for Nautilus Minerals. And this is a person right here. So they're really quite large machines. And as you can see from that head, they can probably do a fair amount of damage to the seafloor. This is a scaled pre-prototype that was developed by the Japanese. And you can see it's a slightly different configuration, but it still crawls along the seabed. And then it uses its head to disaggregate the deposit. Now, because deep sea mining hasn't happened anywhere in the ocean yet, it's really hard to manage or anticipate what management might be needed for the effects of deep sea mining. So one of the ways we decided to try and address this was through using the functional traits of the different organisms to try and determine what their sensitivity would be to different types of mining impacts based on what we know about those mining impacts. So I'm not going to go into too many of the details here, other than to say that functional traits, it's just a different way of looking at your organisms. Instead of looking at its name, so for example, an apple is an apple, you could have different types of apples that are green, red, sweet, crispy, whatever, and you might choose to group your apples by their taste or their color instead of by what variety of apple they actually are. And that's kind of the principal use of functional traits. So instead of using species, comparing species composition between sites, we might look at functional trait composition. So the abundance of highly sensitive organisms in this particular case. So we characterized different organisms using their functional traits. And the outcomes of that was that in general, organisms were more sensitive to the extraction phase of mining, which is where you remove all of the ore from the seabed, than they were to the plumes that were released from the vehicle putting back that return water, or from the actual vehicle moving over the seabed. So different stages of deep sea mining are potentially, organisms are more sensitive to different stages. That's what this shows. And you can see the proportion of taxa that were sensitive to those different disturbances, 2 being very low sensitivity, and then 5 being very high sensitivity. And what you can do, you can then map these sensitivities over the seafloor. So in here, each of the dots or little squiggles are assemblages. So they're samples on the seafloor effectively. And you can then begin to look at where your sensitivities to these different impacts are. These are for the plume. So you can see, for example, where you get the really hot colors are the most sensitive areas. And you can see that they're, for example, they're on the cone of Brothers here. And they're a little bit on the flank of Rumble 2 West. If we go back to Proteus 1, they're up in here where we know we have hydrothermal vent communities, for example. And hydrothermal vent species and some corals and sponges were some of the most sensitive taxa, which was not strictly a surprise. And when we mapped these sensitive areas and then compared them to maps we had of the hydrothermally active and inactive areas, you can see that hydrothermal activity is not just a predictor of assemblage structure, it's also a predictor of how sensitive different assemblages are as well. So now we move into the fun part, the fun part where we talk about environmental management. So we've talked a lot about New Zealand and sort of the environmental setting and some of the biological communities, the maritime activities and the impacts of those activities or potential impacts. How do we then bring all this information together towards an integrated environmental management strategy for New Zealand seamounts? Well, the first step is collecting that existing knowledge. So the things we've talked about today in the environmental setting, things like physical oceanography, biodiversity, so where different assemblages occur, the population connectivity, where you find your fishes of interest in terms of the maritime activities, and also where organisms are most sensitive. We then need to identify knowledge gaps, so where we don't have information that perhaps we might need to make suitable management decisions. And then we somehow need to apply all this knowledge in some sort of framework, and I'm proposing that we use a systematic conservation planning framework for this. So we're going to start with seafloor community structure, and this will make a bit more sense as the slides go on. This information has come from a book chapter that I'm writing with Malcolm, Ashley and Jonathan, so don't worry too much about the references to different sections. But here's the methodology that we propose for providing that biodiversity information on the seafloor community structure that would be needed for management. And there are some steps in here, and these are specifically for deep sea mining. That's the kind of case study that we're using here. And during the process of writing this chapter, we did a lot of detailed looking at the information we have in New Zealand and the information we don't have. And we identified a whole heap of knowledge gaps, which was something that I mentioned in the slide before. I'm not going to talk about too many of them, but I think the three kind of knowledge gaps we had were that of all those seamounts, and on the Kermadec volcanic arc, there are a lot of seamounts, we've only studied very few in detail, and we haven't used the same methodology for looking at all of them either. So in terms of determining a spatial and temporal baseline at the regional scale, we would need to survey more seamounts with the same methodology, and to do that repeatedly over time to figure out your natural environmental variation. And as I mentioned before, most of these studies have looked at assemblages, so parts of the community. They haven't looked at the community as a whole. So we need to do more comprehensive community studies, looking from microbes all the way through to fishes with benthic invertebrates in the middle. And as I said, that in some cases, the same methodology haven't been used for different studies. And in other cases, the same image library for organism hasn't been used. So if we're looking at, for example, these images, and this would be something that during my PhD, I would go through and have to identify all the different organisms in images much like these. You would have an image library that says, right, well, this coral looks like this. And then you can go through and say, right, is it that coral or is it a different coral? If you don't have the same standard image sets to work from, people can call the same coral different things. So that's something we would need to do in terms of knowledge gaps. In terms of important information from seafloor community structures, we saw earlier, we have unique assemblages associated with hydrothermally active areas and hydrothermally inactive areas. And these are things we might want to consider if we go down the route of protected area networks. If we go back and look at our population connectivity, and again, this little diagram of methodology is from the book chapter, so you don't need to worry about it too much in detail, but it shows how we would then determine population connectivity in the context of a new deep sea mining operation, for example. The sort of knowledge gaps that we identified during this process, the three main ones, we only know population connectivity for a very small number of species, really, in the grand scheme of things. And there are many, many species that could be impacted by deep sea mining or fisheries. So you really need to assess a larger range of species with different life histories, for example, and you need to use a range of genetic markers. You could have the same species and you could use a different type of genetic marker to look at its population connectivity, and you would get different answers. And if you want to have a protected area network, for example, that's connected, you probably want multiple lines of evidence to make sure you're protecting the rice. And the other comment was that we need to design our connectivity studies at suitable scales. So in this image, this is a subset of the Gigantidus gladius mussel image I showed you earlier. And we were looking at connectivity within one feature, which is the Kermadec volcanic arc. If you're only looking at managing the volcanic arc, then that's fine. But if you're looking at managing New Zealand as a whole, going back to those early maps, then you'll need to look at other features too, such as the Louisville Seamount chain, for example, and the Chatham Rise. And one of the other take-homes from that work was that we really need to clarify ocean current patterns better to be able to determine the actual routes of larval dispersal and the potential connections, so that we're not just protecting where organisms may have come from in terms of the population, but we're also considering the routes they take to get there. And then looking at the sensitivity work again, this diagram is the same as the previous ones in terms of it's got a methodology that we propose for determining seafloor community sensitivity in the context of mining. Some of the key knowledge gaps were that we don't have functional trait profiles for all of the organisms that you would find in an area, or you have to develop them if you don't have them available at the time. A lot of the time we don't have information on vulnerability traits. We can determine sensitivity, which is fine, but we can't go all the way through to vulnerability without information on life history traits and reproductive traits, which we quite often don't have for deep sea organisms. And all of this is qualitative, of course, because it's based on our interpretation of what deep sea mining disturbances would look like, and that will change as the equipment obviously becomes developed. And so in the absence of actual quantitative information, potential options are a controlled disturbance experiment. So you could actually start to look at how organisms actually respond to simulated deep sea mining instead of hypothesizing how they may respond. And in terms of important take homes from this work, that hydrothermally active areas and hydrothermally inactive areas, so areas that aren't currently active but have been previously, they seem to be more sensitive to deep sea mining impacts than some of the other areas, and also elevated hard substrate areas with lots of corals and sponges are particularly sensitive. So if you're thinking about your protected area networks, these are sites you may wish to include. So we have these elements moving towards integrated environmental management. I know this is a big scary flow diagram. I had to split it into two because it was too big to put in one page in this direction. But this is just to show what a framework for systematic conservation planning could look like if we were thinking about, in this case, our case study is deep sea mining of the Kermadec volcanic arc, but you could also use it for fisheries or other places in New Zealand, or indeed other seabed areas in general. I'm not going to talk about it too much, other than I wanted to point out this early phase that before you even start bringing your data together, if you know you want to do systematic conservation planning, then there's a stage we have to talk to other people, which is the engaging with stakeholders piece here, which I just want us to bear in mind because we will come back to it a little bit later. So we'll have a couple of slides where I can show you where all the things we've just talked about fit into this framework. And then I'll end with some recommendations. So you don't need to worry too much about what's in this slide. It's just to show you that there is a framework where everything could potentially fit together. So focusing on the first half of that framework, you'll recognize these little flowcharts that we looked at in the last few slides. So we're talking about characterizing our seafloor community structure. That fits in up here when we're compiling our biodiversity data in the region of interest for mining. And it also fits in here when we are starting to identify a conservation objective based on what we know actually occurs in the region. And seafloor population connectivity comes in the same sort of places. Then when we start to look at our seafloor community and habitat sensitivity, that comes in under, again, identifying our conservation objectives. For example, if we wanted to conserve sensitive habitats or communities, then information on which habitats and communities are sensitive is important to include here. And then moving on to the bottom half of that flowchart, we then need to think about how all of that information we've just discussed actually then goes into decisions on which areas do we protect and how do we protect them. So obviously there'd be initial phase of reviewing your current achievement of whatever your conservation objectives are, and that involves looking at what you've already protected, which is something that we discussed a little bit earlier. So we have all our benthic protection areas, which are in red on the map. We also have this proposed Kermadec Ocean Sanctuary here. It's not enacted yet. And we have our minerals reserve area here. So then we need to look and say, well, are there additional sites that we need to potentially add to our network? Are sites sufficiently connected for it to be considered a network? Do we need additional sites to actually improve the connectivity amongst sites that we already have? Do we need to change the site boundaries, for example? So things we could do here, we could expand existing benthic protection area networks. We could include habitat-based protected areas, maybe trying to include more of those hydrothermally active or hydrothermally inactive areas, or areas with high density of corals, for example. We then need to apply any conservation actions that we agree on. So this might be the application of the proposed Kermadec Ocean Sanctuary or any additional areas that we decided to include. It could also be imposing any activity restrictions, for example. So you might not be able to do some things within one area, but you may be able to do others. There could be mixed activities allowed. And then you need to manage your selected areas to maintain the conservation value, which is sort of a step a little further down the road. And this diagram was developed in the context of mining, but also you need to be able to link with other sectors for fisheries. So BPAs, for example, these benthic protection areas are only for fisheries, they're not for mining. The minerals reserve area is for mining, not for fisheries. So there needs to be a discussion about how these areas are used. And ultimately, we need to decide, are the current conservation measures sufficient? And how can additional information that we discussed in terms of our knowledge gaps then be gathered to support decisions or improve decisions later to improve the scientific evidence base? And I said I'd talk a little bit about the Kermadec Ocean Sanctuary, and I very briefly will. So this orange area up here, it was proposed in 2015, right after I submitted my thesis. So it's been on the table for a while, but the New Zealand government hasn't finalised what that area will do. So initially it was proposed as a fully no-take, which sounds wonderful. But if you remember on that diagram a couple of slides back, I said that you needed to engage with stakeholders as part of systematic sort of conservation planning. Some stakeholders didn't feel fully engaged in the process, and a full closure here was thought to impact Māori fishing rights. So then there was a concern that actually it's not taking everyone's views into consideration and that it can't go ahead as planned. And as yet it's still not implemented, but discussions seem to suggest that it may be a mixed activity, that it could be some areas are closed to fishing and some areas aren't, and that might be the way forward. But it goes back to why you need your systematic conservation planning process to make sure you protect the correct, the best areas you can, but also involve all the people you need to involve to actually make those decisions inclusively. So in terms of how can additional information support these decisions, well, the answer for scientists is always, let's go do more science. We need more information. So the story doesn't end for New Zealand here. There are ongoing initiatives, and I've raised a couple here. If for people who are interested in ongoing New Zealand work, one of which is the Global Seamounts project, and you'll see there's a lovely picture of Malcolm Clark here. And if you're interested in the New Zealand aspect of that project, I'd strongly recommend that you contact Malcolm because he's very heavily involved in that. There's also the Challenger 150 programme, which will involve work from New Zealand as well as many other places. And actually the next speaker in the Interridge series, Anna Hilario, I believe, is going to talk a bit more about Challenger 150. And I think, if I'm correct, that it was a proposed UN Decade programme, and it's actually been formally kind of accepted by the UN as the UN Decade programme, which is very exciting. So that's it. That's all I have for you today. Thank you very much for your attention, and I'm very happy to answer any questions you may have.